Hello everyone. This is Chapter 8, Bivariate Correlational Research. This is a chapter that I really enjoy. I'm a statistician, so I like talking about stats. But for you, we're not going to go into a ton of depth. We're just going to introduce these topics. If you've taken a stats course already, then you're going to recognize some of these things. If you haven't, this will help you as you go into those courses. So this chapter, we're going to be talking about bivariate correlations. Bivariate just means two variables. We're also going to be interrogating association claims. So we're going to look at what makes a good association claim and what makes it overreaching or a poor association claim. So when it comes to bivariate correlations, that's an association that involves exactly two variables. Those two variables can both be continuous, or we can have categorical variables in there. This is an example of some data that we could do a bivariate correlation with. So each person is a single row. So person A through ZZ. And each person has a score on well-being and the percentage of conversations rated as deep talk. So person A scored a 4.5 on well-being and an 80 on deep talk. Some other examples, we could ask, where did you meet your spouse? And your current marital satisfaction rating. So unlike the ones that we've talked about so far with experiments, we're not assigning whether they meet their spouse online or offline. This is something that just happens, right? So for me, I met my wife offline. No experimental control happened. That's just a measure of me. And then my marital satisfaction would have a number. And we can look at the relationship between where we met and marital satisfaction rating and get a correlation between those. This is another example. We won't go into detail, but the idea is still the same. Each person is a row and they have exactly one score for this measure and one score for this. So when we introduce bivariate correlations, we're going to be describing associations between two quantitative variables, and we'll be describing associations with categorical data involved. So this first one is quantitative variables or continuous variables. So we have the well-being scale, it's z-scores, that means it's just standardized. So a negative score means you're below the mean and a positive score means you're above. And then we also have a score for our percentage of deep talk. So each dot is a single person. So this person right here scored pretty high on well-being and pretty high, one of the highest on percent of deep talk. This person right here was somewhere around the middle on both. So overall, we can look across the individuals and see a relationship between these two variables. This line tries to highlight that. It's a positive relationship. In general, when I have a higher well-being score, my percentage of deep talk is usually higher. But that's not always true. As we can see, this person has a really high percentage of deep talk and not a super high score in well-being. So this one would be maybe a moderate, small to moderate correlation. Cohen gave some guidelines about what different values of R, it's Pearson's R, it's a correlation measure. It's how we quantify the relationship. And what he said was 0.10 is about, it's a smaller, weak relationship. 0.3 is medium or moderate. 0.5 is large or strong. This is totally dependent on your area, but these are just some rough guidelines to help you understand when you see a correlation reported, roughly what that correlation, how strong of a relationship that means. We also can look at associations when one variable is categorical. So this one is the marital satisfaction. and where you met online or offline. And so we see we have data points across marital satisfaction for online and offline. If we just look at the means of the two groups, online has slightly higher than offline here. But we can see there's a lot of overlap between the two groups. And when I talk about the mean, if you haven't taken statistics, it's just the average, the arithmetic average. So it's the average of all these points would be right here. And the average of these points would be right here. So the question is, what makes a study correlational? Well, it is having two or more measured variables with no experimental manipulation. So association claims basically should always be in this situation. We should not have any real causal claims here. We're just measuring variables and looking at their relationship. We're not doing any manipulation. We can potentially get causal claims, but only in very rare circumstances. So when it comes to interrogating association claims, we can look at construct, statistical, internal, and external validity. So construct validity, that would be asking how well was each variable measured? Are they really well measured? Do they measure what they should measure? Statistical validity asks how well do the data support the conclusion? So do the stats support what the researchers are claiming? Internal validity, it asks, can we make a causal inference from association? So if the correlation is really strong, then maybe we can start to go towards some causal inference with a lot of cautiousness, I would say. So internal validity is often not emphasized in association claims because it often can't lead to this causal inference idea. External validity, on the other hand, says to whom can the association be generalized? So in the last chapter we talked about a good sample. If we have a good sample, then our association can be generalized to that sample. If we have a really poor sample, then our generalizability is probably really low. So I mentioned effect size earlier when I was talking about the correlation itself. Effect size just describes the strength of an association. So for example, there's A and B plots here. Which one has a stronger association? B would be the one with a stronger association because the points are more tightly together in a line. If you were to draw a line through these points, the points would be very close to that line. This one, with the same scale, so X is the same and Y is the same in both, this one, the points are more scattered around the line. So the effect size in B would be larger than A. So consider this. So height at age 2 and height at age 18 turns out correlate pretty strongly. The line right here is what's called the prediction line. It's a line that tries to connect to fit the dots the best. So we actually can use this line to get a prediction measure. So we could say Hugo was, when he was 2, was 83 centimeters. So we could draw that up to the line and we could say when he gets to be 18, we expect his height to be 172 centimeters. In fact, when he turned 18, he was 170. It's a pretty good guess considering the wide range of possible heights that are in this data. So when the association is really strong like this, it helps us be able to predict an outcome better. On the other hand, the mother's height and the son's adult height, they do correlate, but less strongly. So this is the prediction line for those data. And in this case, Hugo's mom is 163 centimeters. So we could draw the line up to there and then follow it over. And it would predict that his height was 174. It's a little less accurate than this one, but still a good guess considering the range of heights that we see in this data. So across these two examples, this one has a stronger correlation. It has a bigger effect size. When we have a bigger effect size, that tells us that this information is going to tell us more about this information than if it was a smaller effect size. So here, this information is very good at telling us roughly what the adult height will be. This one is okay. It's not as good because the effect size is smaller. So that whole idea, when everything else is equal, a larger effect size is usually considered more important than a small one. But of course, in science, there's always some exceptions. For example, in a medical study on heart attacks and taking aspirin, a small, where the correlation, the R, was 0.03. And if you remember, 0.1 was considered small. So this is even smaller than that. Was actually considered extremely important because the number of lives that it would save. There were 22,000 participants in the study, 11,000 in the aspirin group and 11,000 in the placebo group. There were 85 fewer heart attacks in the aspirin group. That's a lot of lives saved for a small effect size. So the effect was really small, but that still saved 85 lives. So it still isn't maybe the best thing to stop a heart attack, but it clearly does work. It helps. So even though there may be other important effect sizes to consider when it comes to heart attacks, this is not an unimportant one just because it's small. So the next question is the correlation statistically significant? If you haven't taken a stats class, that's okay. The idea of statistical significance essentially just means we believe that this effect is actually happening in the population, not just in our sample. That means we understand that our sample isn't going to be perfect, that there's going to be some unique things about the sample. And so any effect that we see in our sample could just be our sample and not in the population. So we use statistical inference to be able to make that call. You'll learn more about this in a stats class. You'll understand what tells us that yes, it's statistically significant or not. But the idea is when it is significant, that is when we claim that what we are finding in our sample would also be happening in the population. When it's not significant, that just means we don't know. It's an inconclusive result. So if someone has a null result, which means they didn't have significance, they shouldn't be claiming that there's no effect in the population or that there is an effect. Basically, it's just our data couldn't tell us that answer right now. In all of this, these three things are usually what researchers are going to focus on in a study. You have the effect size, so how big is the effect? How big is our sample size? And significance. Those actually all relate to each other. So when we have a really big effect size, we actually don't need as big of a sample size to find it significant. When we have a really small effect size, we actually need more people to find it significant. I like to think of this as like a detective. You're looking for evidence of an effect somewhere. If the effect is really obvious, it's really big, you don't need a whole team of people to help you find that effect. So you can use a smaller sample size to find it. If you're looking for something really small, then you're going to need a team of people to help you find it. So the team is like the sample size, and what the detective's looking for is the effect size. So with a big sample, we actually can find small effects. So like the example I was talking about with the medical study with a small effect, that one had a really big sample of 22,000 people. So it was able to find a really small effect and still find it significant. So they could still conclude that the effect actually exists in the population. If they didn't have that big of a sample, they wouldn't have been able to find that. So if you are interested in learning more about statistics, reach out to me. Take the Psych Stats class if you can. There's other classes that you can take to learn more about this. Stats is really the language of science, so you do need to understand it somewhat if you want to do research. But it doesn't mean you have to be a statistician. So a question we always need to ask when it comes to looking at our data and getting an association is, are there any outliers? Outliers in small samples can really affect your results. So for example, this person, it's the same person as this. In this sample, it's smaller. It's going to drive the results more. The line is going to look more like this in this sample, and it's not going to drive it as much in this sample. So this extreme score is way far away from everybody in terms of this variable. And it's the same as here, but this data, it's more clear. We have more individuals to know this is the normal range. This person is kind of far away from that. So anytime we're looking at our data, we need to consider, are there outliers? Now this person, it doesn't mean we need to take them out of our analyses. It's just something we need to understand, that that person could be influencing the results somewhat. However, we might look at this data and we might say, well, the highest score you actually can get on this variable is a 10, so this must have been miscoded. That would also be good to know. So outliers can also be something that, oh, we made a mistake, let's correct that. And we can fix it and then run our analyses. Another thing to consider is whether there's a restriction of range in one of your variables. So these two plots show college grades, their GPA, and their SAT score. And you may hear that SAT score doesn't predict college grades very much. That's often because of a restriction of range. So in this example, the correlation between SAT score and college grades are somewhat positive, but not super strong. In this data, it is 0.33, so a moderate effect. But why would it be cut off? Because often colleges decide to admit somebody if they get a score, let's say, above 1200. So we don't even know the grades here. But if we could get those grades, let's say these were those grades, and look at the correlation between SAT score and college grades, then the correlation is much stronger. It's 0.57. That's a large effect. So even though it feels like SAT scores do not predict college grades, it may just be because it's a restricted range, and so we don't get the full picture of how well the SAT score is actually doing at predicting grades. Another thing to consider is whether the association is curvilinear. Curvilinear just means a straight line doesn't fit through it very well. Instead, a curve would fit through the lines better. So in this example, it's age in years and the use of healthcare system. So when we're really, really young, so babies, toddlers, newborns, we use the doctor quite a bit. But during our teen years and as we're getting older, generally speaking, we're more healthy. And so we don't need to use the healthcare system nearly as much. But as we age, we use it a little bit more. And then once we're in our senior years, we're starting to use it a lot more again. So this example, if we were just trying to look at a regular correlation, it would try to fit a straight line here, and the line would just be flat. There'd be essentially no relationship. So we'd want to understand that this is actually a curve. So in any claims that you see, if you think, well, maybe that could be a curvilinear relationship, and all they're looking at is a correlation, they may have missed that. It's something to look into. So we're going to consider internal validity as one of the 3 causal criteria. When we talk about internal validity, we're going to get this potential third variable problem that's mentioned on this slide. The 3 criteria are, first, covariance. That just means are they correlated? Do they move together? If one's higher, is the other one higher? Or is one higher, the other one lower? Or is it just there's no relationship? If there's covariance, that's the first criterion. Second is temporal precedence. We need to know, did A happen before B, or did B happen before A? If A happened before B, then A may be a cause of B. If B happened before A, then it could be that B is a cause of A. The third thing is internal validity, and this is about the third variable problem. Is there a third variable we'll call C here that is associated with both A and B independently? So this variable that could, in essence, be causing both A and B to happen. If there is a plausible third variable, then we can't infer causation. So one of my favorite examples of this issue is there's a strong relationship between chocolate consumption and car accidents. So we could assume, well, maybe when people eat chocolate, they just get so drunk on chocolate that they cause car accidents. Or maybe they're eating chocolate in the car somehow and the car is causing them to choke and so they crash. But what it turns out is it's actually this third variable. And the third variable in this case is what is the date. Essentially is it a holiday? If it's a holiday, we're more likely to eat chocolate. And on holidays, we're more likely to travel. And so because it's a holiday, we're going to eat chocolate and travel more. That is actually what's happening. When we travel more, there's going to be more accidents because there's more cars on the road. So in this case, A didn't cause B and B didn't cause A. It was actually a third variable that was causing both of them. Another thing we should consider is external validity. This asks a question, how well does our results generalize to the population? Sometimes that's very important to a study, other times maybe not so much. The idea, though, is to look at how good is their sample. What's interesting is this figure shows 2 different groups, so you have young and old. Even though the young score much higher on time spent multitasking and their ability to multitask than old people in general, turns out the relationship between the two, it's still the same. There's a negative relationship in this group, and there's a negative relationship in this group. So sometimes, even though one study is just about young individuals, it may generalize to some degree to older individuals. But that is something that we should look at empirically, so we should look at data and see. So another thing to consider is moderating variables. This example I have here, I think, is well illustrated in these plots. What this is telling us right here is that a city's degree of residential mobility moderates the relationship between local sports team success and attendance at games. So each dot represents one Major League Baseball season. The Arizona Diamondbacks are the one in the red, based in Phoenix, a city with high residential mobility. And this pattern shows that people are more likely to attend games when the team is having a winning season in Arizona. Pittsburgh, a city with low residential mobility, it's actually the opposite. The pattern shows that Pittsburgh Pirates, the Pittsburgh team, fans attend games regardless of how winning the season is. It's slightly negative, but mostly not much of a relationship. The location is what moderates the relationship between percentage of wins and attendance. So a moderating variable just changes how a relationship looks depending on its own level. Another example to consider of this is the effect of, or the relationship between eating ice cream and enjoyment. The relationship between those two might be moderated by the type of topping you choose. If it's mustard, you're probably not going to like ice cream very much. So the relationship between enjoyment and ice cream is not going to be very high. If it's fudge, if you like fudge, then you may really enjoy the ice cream. So the amount of ice cream you eat is more positively related to your enjoyment. So that is it for Chapter 8. Please reach out if you have questions. We didn't go into a ton of depth into the statistical nature of correlations, but hopefully you understand what the claims are and what they mean and the things that go inside of a correlation, including effect size and significance.